Hello everyone, it is a pleasure to share with you our article published in iMeta, Lactobacillus reuteri-mediated dietary xylooligosaccharides enhance jejunal cell survival via suppression of oxygen-dependent apoptotic processes in a pig model. Gut microbiota balance is crucial for mammalian growth and development. Prebiotics such as functional oligosaccharides can regulate gut microecology by promoting probiotic proliferation. The traditional belief is that functional oligosaccharides passively pass through the small intestine and ferment only in the distal colon. However, emerging evidence indicates they can directly modulate epithelial cells, enhance intestinal barrier function, and activate immune responses. This paradigm shift calls for a re-evaluation of their scope and mechanisms in gut regulation. Our study has three highlights. First, we confirmed the mechanism by which xylooligosaccharides inhibit epithelial cell apoptosis via reshaping jejunal microbiota composition and host energy metabolism. Second, we identified Lactobacillus reuteri as the core microbiota mediating xylooligosaccharides-induced jejunal benefits, with glycosaminoglycolic acid as the key anti-apoptotic effector metabolite. Third, these findings provide new insights into the molecular mechanisms of crosstalk among oligosaccharides, gut microbiota, and epithelial cells. We fed xylooligosaccharides to 28-day-old weaned piglets for 28 days. The results showed significantly increased villus height in the jejunum and ileum of the xylooligosaccharides group, providing a stronger structural foundation for nutrient absorption. Further investigations revealed that xylooligosaccharides improves epithelial cell survival not by promoting proliferation but by upregulating the anti-apoptotic protein BCL2 and downregulating the pro-apoptotic proteins caspase-3 and BAX. To elucidate the mechanism, we profiled the jejunal mucosal proteome. Enrichment analysis of the differentially accumulated proteins showed significant alterations in energy metabolism pathways, particularly the suppression of oxidative phosphorylation and tricarboxylic acid cycle enzymes, indicating reduced cellular oxygen demand. Given the critical role of small intestinal microbes in nutrient sensing and digestion, we analyzed the microbial composition of the jejunal mucosa and lumen. Xylooligosaccharides significantly altered the alpha and beta diversity of the mucosal microbiota, enriching beneficial bacteria such as Lactobacillus spp. Lactobacillus reuteri as the core species driving the increased abundance. Using an Escherichia coli-induced villus injury model in piglets, we further evaluated the effects of Lactobacillus reuteri supplementation on jejunal villus height and nutrient utilization. Lactobacillus reuteri treatment effectively reduced Escherichia coli-induced weight loss and alleviated villus atrophy. Importantly, it inhibited caspase-3 and BAX, upregulated BCL2, but had no significant effect on the proliferative marker pCNA. We hypothesized that the xylooligosaccharides-enriched, Lactobacillus reuteri-centered microbiota attenuates apoptosis via microbial metabolite epithelial cell interactions. Non-targeted metabolomics of jejunal digesta revealed significant changes in bile acids and other metabolites. Multiomics integration confirmed metabolites as key mediators linking microbiota and epithelial function. Targeted analysis showed both xylooligosaccharides and Lactobacillus reuteri increased glycosaminoglycolic acid levels. Moreover, in vitro cell experiments confirmed that low-dose glycosaminoglycolic acid effectively inhibits hypoxia-induced apoptosis of intestinal epithelial cells. Our study reveals a previously unrecognized function of indigestible xylooligosaccharides in the proximal gut, enriching Lactobacillus reuteri to inhibit apoptosis, increase villus height, and enhance nutrient transport, thereby improving growth performance in weaned piglets. Mechanistically, Lactobacillus reuteri removes energy metabolism and reduces oxygen dependence, promoting epithelial cell survival under hypoxia, closely associated with glycosaminoglycolic acid accumulation. These findings significantly advance the understanding that oligosaccharides function beyond hindgut fermentation, highlighting xylooligosaccharides' role in host microbiota interactions and jejunal microbiota's contribution to epithelial protection and gut homeostasis.